Hello everyone, welcome back to BioSchool. This is Dr. Gayatri. In today's session, I'm going to explain you questions from 31 to 50 of Part B of CSIR NET Examination 2020. And these questions are from morning session. I have already uploaded questions of Part B of evening session and question number 1 to 30 of morning session. You'll get the links in my description box. Please do watch. If you are a life science student and preparing for any competitive examination, then I will recommend you to go through all these videos because it will definitely help you a lot and the related type of question you may get in your examination. So let's proceed. Question number 31. Which one of the following snRNA given below base pairs with 5' splice site of pre-mRNA? So this question is from spliceosome mediated splicing. So here you can see this is exon 1 and this is exon 2 and this is intron. 5' end has GU and 3' end has AG and A, this is the branching point. Okay, this is the branching point. So in this question it is asking which one of the snRNA given below it base pair with the 5' splice site of mRNA. So see here the 5' splice site. First, the U1 sRNA it binds, and to the branching point U2 snRNA binds. When the U1 snRNA it get released, then that site is occupied by U6. Okay, so in the option there is no U6, only U1. Okay, the correct answer is U1, which base pair with the 5' splice site of pre-mRNA. Question number 32. Mycorrhizal fungi are associated with a large variety of plant species. The diagram below shows the cost-benefit curves from individual plants with or without mycorrhizal fungi associated with the roots across a soil nutrient concentration gradient. Which one of the following option best describes the association between plant and mycorrhiza when soil nutrient concentration are high? Okay, so this is the plant size and this is the soil nutrient concentration, and you see this graph, the solid line, it represents the plant without mycorrhizal fungi, and this is the dotted line, it represents the plant with mycorrhizal fungi. So when the soil concentration is high, then the plant without the mycorrhizal fungi show the benefit. Okay, this benefit to the plant. But when the plant is with mycorrhizal fungi, then that is harmful. That cause harmful. Okay, so it is which type of association? So one, if the plant it is without mycorrhizal fungi, then it get benefit. If it is with mycorrhizal fungi, then it get harm. So this type of the association is plus and minus. If the mycorrhizal fungi present, then that cause harmful. If it is absent, then that is beneficial. So let's see, it is a parasitism type of interaction, because parasitism is the plus minus, mutualism it's a plus and plus because both will get benefit. Competition in which both the species they get harm. So it's minus minus. Commensalism in which one species it will get benefit without affecting other species, so it is the plus 0. But here if the plant it is with mycorrhizal fungi, then it get harm. If it is without mycorrhizal fungi, then it get benefits. So this is the parasitism, which best describes the association between the plant and mycorrhiza when soil nutrient concentration are high. Correct answer is option 1. Next question. In a cloning experiment, a DNA molecule isolated as PST1 fragment was inserted into the PST1 site of a cloning vector that is 3 kb in length. Cloning vector has 3 kb in length. Digestion of conformed clones with PST1 resulted in the appearance of a single band. It formed a single band corresponding to 3 kb in agarose gel. Based on the information What is the probable size of cloned fragment? Okay. The cloning factor, it has the size 3 kb in length. Digestion of conformed clone with PST1 resulted in the appearance of single band. It appeared as a single band that is correspond to the 3 kb in agarose gel. So based on this information, what is the probable size of cloned fragment? So the cloned fragment size and the cloning factor size, it is similar. That's why it forms the single band which is correspond to 3 kb in agarose gel. Otherwise, two different bands will be formed. So what will be the size of the cloned fragment? It is also 3 kb in length. That is correct answer is option 3. Question number 34. Which one of the following is not an extracellular matrix protein? So, the keratin. Keratin, it is present in the hair, nails, and we know that keratin, it is a cytosolic protein. It is not an extracellular matrix protein. Okay. And the other three, that is laminin, collagen, fibronectin, all these three are extracellular matrix protein. So the correct answer is option 1, that is keratin. Keratin, it is a cytosolic protein. Question number 35. This is very simple question from animal physiology. It is asking about myosin molecules that assemble into bipolar filament in the muscles are, in muscle which type of myosin is present? That is myosin 2. Okay. Correct answer is option 2. Okay, let's move to next question. Which among the following states has highest forest cover in terms of percentage of geographical area? So it is asking which state has highest forest cover in terms of percentage of geographical area. That means you have to know out of these 4 states which state has highest forest cover in terms of percentage of geographical area. This question is out of box and it's a little bit hard to answer, and the correct answer for this question is Uttarakhand. Okay, Uttarakhand, it has the highest forest cover in terms of percentage of geographical area. If it is asking about which of the state has highest forest cover, that is Madhya Pradesh, but it is asking the percentage of geographical area. So correct answer is option 2. Question number 37. Which one of the following genome is most appropriate to determine hybrid in plants and animals? Okay. Which one of the following genomes is appropriate to determine hybrids? Okay. In plants and animals. If there is an animal, in animal, chloroplast is absent. So option 3 and option 4 both are wrong. Okay. Then the correct option, it will be either 1 or 2. Which one of the following genome most appropriate to determine hybrid? So it's obviously the nuclear genome. It will help in determining the hybrid in the plants and animals. So the correct answer is option 1. Question number 38. Which one is not the function of P cell, that is principal cell, in collecting ducts? Okay, the collecting duct, it has the two type of cells. One is known as the PC cell, that is the principal cells, and other cell is known as the intercalated cells, that is the IC cells. Okay, so the function of P cells is in sodium reabsorption, potassium secretion, and reabsorption of water molecule, whereas the intercalated cell, it help in transportation of acid and base. So here in question it is asking which one is not the function of P cell. So hydrogen secretion, it is not the function of P cell, it is the function of IC cell, intercalated cell. Okay, and all these three, like sodium reabsorption, potassium secretion, and water reabsorption, are the function of P cell. Okay. And this is not the function of P cell. So this is the function of intercalated cell, IC cell. The correct answer is option 4. Question number 39. Examples of antibiotic resistance highlight important features of natural selection. Okay. Which of the following statements is not true? It is asking which one is not true. Okay, let's see. Evolution by natural selection is progressive. It makes individual better. Is the wrong answer, because the natural selection, it has no foresight, no direction. So we can say that the natural selection, it is progressive and it will make better individuals. So this one is not true. And let's see the other options. Natural selection acts on individuals, but it is population that changes with time. Yes, its population changes with the time. This one is correct. Natural selection does not cause genetic change in individuals. This one is true. Natural selection acts on phenotype. This one too is also correct. So the wrong one is, or option 1 is not true. The correct answer is option 1. Let's see question number 40. Which of the following phytopathogens has predominantly necrotrophic mode of colonization? Necrotrophic means dead. Okay, dead type of colonization. And this question is easy for the botany students, but it's hard for zoology students. So the correct answer for this question is Irvinia. This is so the necrotrophic mode of colonization, whereas the other three that show the parasitic mode of colonization. So for this question, the correct answer is option 2 is Irvinia species that show the necrotrophic mode of colonization. Question number 41. Identify the plot that depicts the change in metabolic rate of an endotherm with respect to change in environmental temperature. Okay, if the environmental temperature changes, then also the metabolic rate is changes in order to maintain the equilibrium of body. So let's see the graph. This graph, this is wrong because it shows the constant line. Obviously the metabolic rate, it will be changed with change in the temperature. So this one is wrong. And option 1, it shows that when the temperature is cold, okay, or in the 0 to the 10 degree temperature, then it shows the metabolic rate is lower. If the temperature is cold, then what will happen? The metabolic rate, it will be high in order to generate the heat. So this one is wrong. Option 3, let's see. Yes, if the temperature is in the cold climate, the metabolic rate of body increases in order to generate the heat. Okay. And when the body temperature in endotherm, we have the 37 degrees Celsius temperature. So in this temperature, the metabolic rate, it will be nearly constant. Okay. But when the temperature it raises like 40 degrees centigrade, then also the metabolic rate will start to increase in order to cool the body. Okay. In order to maintain the temperature. So this one is correct for endotherms. So, and this graph, it is for the ectoderms. Okay. This graph is for ectoderm. The correct answer is option 3. Okay. Question number 42. Mutation is essential for genetic variation. Yes, mutation, it causes the variation. Which one of the following event can lead to variation amongst the gametes produced by male Drosophila melanogaster? Crossing over does not occur in Drosophila melanogaster males. We also know that crossing over, it does not take place in melanogaster males, but it is given in the question to help you. So mutation that cause the maximum variation. Mostly the variation is caused because of the independent assortment. Also recombination that cause the variation. But here in question it mentioned that crossing over, it does not take place in melanogaster males. So there is no recombination. So which of the following event that will be cause the variation? That will be the independent assortment. Okay, because the segregation, it does not cause the variation. Also imprinting and the recombination, it causes the variation, but in Drosophila melanogaster male, the crossing over or the recombination is absent. The correct answer is option 4, that is independent assortment. Okay. Question number 43. Which one of the following options represents a series of amino acids with the decreasing pK value of their side chains? Okay, it is asking about the side chain or the R chain, and it asking in the question, it asking the series of amino acid with decreasing pK value. Okay, so if the pK value is high, Then it has amino acid as more basic. Okay. If the pK value it decreases, then basic property it is also get decreased and acidic property increases. So in the option, which amino acid has highest basic properties? Okay, the basic property it will be highest in arginine, then followed by the lysine, then cysteine and histidine. The correct option is option 1. So here you can see in the side chain also the pK value has given. You can see for the arginine the pK value it is 12.48. Okay, then followed by lysine. For lysine it is 10.79. Then cysteine, for cysteine it is 8.33. Then histidine it is 6.04. Okay, so in decreasing order the amino acid sequence is arginine, lysine, cysteine and histidine. Okay, the correct answer is option 1. Okay, the next question, which one of the following sets of terms is matched correctly? Okay, let's see the option. Fluorescence microscopy. Use of dyes that absorb and emit lights of same wavelength? No. Absorption takes place at lower wavelength and emission takes place at higher wavelength, not at the same wavelength. So this one is wrong. Confocal microscopy. Use of pinpoint illumination and detection of apertures. Yes, this one is right. Option 3, confocal microscopy. Use of pinpoint illumination and detection apertures. Both the options are same, so the correct answer it is both, that is option 2 and option 3. It is a printing mistake. Option 4, gas chromatography, used for analysis of non-volatilized carbohydrate molecules. No, it is used for analysis of volatilized carbohydrate molecules. So this one is wrong. The correct answer is option 2 and option 3 because both have the same sentence. Question number 45. Which one of the following proteins produce a dark blue or purple color in the presence of 5-bromo 4-chloro 3-indole phosphate and nitro-blue tetrazolium? So which of the enzyme it will remove the phosphate? The phosphate it will be removed by the phosphatase. So the correct answer is alkaline phosphatase. Okay. Option 2. If the phosphate, when the phosphatase it remove the phosphate group, then it will produce the dark blue or purple color. So let's move to the next question. Okay. Cytoplasmic male sterility in plants is caused by mutation in mitochondrial genome. Cytoplasmic male sterility can be restored by a nuclear gene restorer of fertility, that is RF, which is a dominant character. Okay. If male sterile pea plant is pollinated by a fertile male pea plant with RF in heterozygous condition, the progeny obtained will have, male sterile pea plant. Sterile pea plant, that means it will be in the recessive. So take it as a AA. This one is male sterile pea plant is pollinated. Pollinated means it considered it as a female. Okay, consider this one as a female. It is pollinated with fertile. This one is sterile. This is fertile. Fertile pea plant. It is present in the heterozygous condition. Okay. Heterozygous condition means capital A, small a. Then the progeny. What will be the progeny? Progeny, it will be capital A, small a, and another one will be small a, small a. Okay. So this one, this progeny is fertile, and this progeny it is sterile. So 50% of the progeny are fertile and 50% of progeny are sterile. If you have any doubt, then please comment down. I will try to reply. So the correct answer for this question is option 3. It is 50% of the progeny for fertile and 50% male sterile. Question number 47. UV induced DNA damage. Okay. Ultraviolet induced DNA damage. It will form the thymine dimer. Causes advancing replication forks to stall. So if UV radiation is present, then it will cause the thymine dimer. If the thymine dimer it forms, then it stops the replication fork. To avoid collapse of these stalled replication forks, the cell uses, okay, it will use the non-homologous end joining. Non-homologous end joining, it is used when there is a double stranded DNA break, but here there is no break, so this one is wrong. Lesion bypass, yes, the lesion bypass. If there is a thymine dimer, it is repaired either by thymine dimer direct repaired by the presence of enzyme photolysis or by the nucleotide excision repair method. But here there is no mention of nucleotide excision repair and the direct repair of thymine dimer by the enzyme photolysis. So the correct answer it will be lesion bypass. Mismatch repair. Mismatch repair it is used for repair when there is a mismatch in base pair. This one is wrong. In base excision repair only the single nucleotide it is removed. Okay. So this one is wrong. So how can it will be corrected or avoid the collapse of the stalled replication fork? Only by the lesion bypass. Okay. The correct answer is option 2. 48. In the context of plant breeding and genetic engineering, which one of the following statement is correct? Okay, let's see the option. To achieve high expression level of heterozygous gene in a transgenic plant, a promoter of bacterial origin is often used. Why for the plant gene expression the bacterial origin promoter will be used? Bacterial origin promoter is not required for the plant gene expression, so this one is wrong. Option 2. F1 progeny derived by crosses between inbreed lines with low genetic diversity is more likely to show heterosis. In heterosis, the F1 progeny it has better fertility and the development than the parent. So it will obviously show the high genetic diversity. So it has the low genetic diversity. So this one is also wrong. In agrobacterium mediated plant transformation, always a single copy of transgene is inserted in the host genome. Most often, multiple copies is inserted, not the single. This one is also wrong. Qualitative traits are typically characterized by discontinuous phenotypic variation. Yes, qualitative traits like you can tell that plant size it is a tall or dwarf. Okay, so this type of the characteristic there is a so the discontinuous variation. Okay, while the quantitative traits are generate continuous phenotypic variation. Yes, so this one is true. So the correct answer is option 4. Question number 49. Which one of the following approaches is incorrect in classifying organisms from kingdom Animalia? So this question is for zoology students. So let's see the options. Spicules were used as primary criteria for classification of phylum Porifera. Yes, the spicules are the characteristics of phylum Porifera. This is correct. Protostome were classified into two major lineages, Lophotrochozoans and Ecdyozoans. So this one is correct. Hemichordates were placed in same superclass as echinoderms because both have ciliated larvae. Echinoderms they don't have any ciliated larvae, so this one is incorrect. Ambulacrians included echinoderms and hemichordates. This one is correct. Ambulacrian that is included both in echinoderms as well as in hemichordates. So incorrect option is option 3. Okay, the correct answer is option 3. Okay, the last question. If you require to generate energy, okay, energy, energy in T cell, which of following condition you see? So what is energy? Energy in the T cell means it is immunologically unresponsive. Okay, when the T cell it is encountered with an antigen on the primary signal, then so what is the energy? Energy means it is immunologically unresponsive. Okay, intrinsically it is inactive. It cannot produce any response. So when the T cell it encounter with any antigen and it becomes unresponsive, then that is called the energy. And that energy it can be removed when the T cell it receive another signal. When it receive the second signal, this second signal it rescue the T cell from anergic state. But here in question it asking, if you are required to generate energy in T cell, you have to make the T cell unresponsive. Okay, it cannot produce any response. So which of the following condition will you see? Stimulate naive T cell with antibody that bind both T cell receptor and CD8. So if both the signals are present, then it is responsive. So this one is not correct. Stimulate naive T cell with antibodies that bind only with the T cell receptor. Okay, there is only signal molecule, only one, the fast signal molecule. So when it is encountered with the antibody or antibody when bind to the T cell receptor, then that cause unresponsive immuno. So that cause energy in T cells. So this one is correct. Stimulate naive T cell with antibodies that bind both T cell receptor and CD20 in presence of interleukin 4. No, both the signals are present, then T cell it will be responsive, immunoresponsive. Permit naive T cell interaction with activated macrophage in presence of interleukin 4. This one is also wrong. The correct answer is option 2. Thank you so much for watching this video. Please go through all the videos of BioSchool. If you like this video, then please do share and subscribe my channel and press the bell icon to get regular updates. Thank you so much.